The change in scale couldn't be more dramatic. So we're going from space into microscopy. Before I start, I have to give a quick introduction. So I joined Oxford little than 2 years ago, and before that I worked for GE Global Research in upstate New York. And if you followed the news recently, GE just set up a whole new business called GE Digital that is essentially just focuses on data analytics for its own businesses. I was somewhat involved in the setup of the precursor, which is now the entity called GE Digital. And I want to talk a little bit about versus capability versus utility. So and I've essentially been on either side of the fence. Sometimes it's very easy to generate large data sets. And one doesn't really have the right questions. And sometimes one has the right questions, and finding good data is suddenly very, very difficult and expensive. So I'm talking a little bit about robotic imaging platforms. I'm going to tell you about a process we developed for molecular pathology at GE. So this is all work that has not been done in Oxford. This is sort of massive. We can essentially generate massive multi-channel data sets, not as massive as the sort of astronomy images you've seen before. But now where I'm now, and you ask specific clinical or specific biological questions, and you actually start assembling data set, it's very, very laborious and very expensive to get to the data volume you really need. So I give you an example out of the area of toxicology screening. The so-called zebrafish is a transparent model organism that transforms depending on certain toxicological states. And until recently, a lot of measurements had been taken by hand. And as you can imagine, if you want to basically screen thousands of compounds, you have to really worry about automation and automated data analysis. So what we did, basically fueled by a development of analyzing, faces with models and analyzing facial expressions, we developed an atlas for zebrafish that essentially has all the anatomical information you need incorporated in the atlas. And you can, for instance, measure the eye, the ear, the notochord length, and essentially the computer will do that all automatically. And there are certain measurements toxicologists came up with, and they can be readily incorporated in something which is called basically a multi-resolution grid. And you can essentially also run genotype studies because you can effectively add all measurements you're interested in to that grid. So from that idea, we went all the way to basically building a piece of analysis software that can essentially analyze images that are being taken in well-played format. So for those of you who have never seen this, is essentially like an egg carton, and in every little box you run its own experiment, and you can have up to 384 wells on a plate, and then you image whatever plates where you have certain experimental conditions. So that's one example where we essentially went from one example, basically one atlas, and can now analyze a whole plate with a push of a button. So we basically were able to really increase the throughput of these kinds of studies by large factors, probably by 100 or 200, because we essentially, before you had basically postdocs that were trained in zebrafish anatomy and did all the various different measurements. Now in a very different example, and a much longer and more expensive project. So the idea here was to image as many as to 100 different proteins in a single tissue section. So the way we approached this problem, we essentially wanted to develop reagents, algorithms, and systems to facilitate this process. Now, in the interest of time, I really can't go into the chemistry, but essentially what you do is you label an image with so-called DAPI. DAPI essentially colors the nuclei, And then you have one functional marker, that's basically the yellow channel. You can chemically deactivate that stain and can repeat this 80 to 100 times. So essentially imagine, like in most microscopy, if you have like 4 laser channels, that's pretty much the end. And now you have an image that has 80 different channels. So there's basically a bunch of technology needed to actually form these data sets. And here, just to give you an example, this is the DAPI channel. These are basically the nuclei. Then you basically color the cytoplasm and the membranes. And then you do something similar than in the zebrafish example. You generate like a map, an anatomically meaningful map, and you use that map to then quantify basically the protein uptake. Because these images, because they're so complex, they're not really human interpretable anymore. So what you get is a high content signature for tissues. Essentially, you have every cell has a number, every cell has parts, nuclei, cytoplasm, and membrane, and then you can have, up to like 200-300 measurements per cell. Okay, and now you're trying to do this. Now you're trying to use this technology in meaningful studies. So we at GE we did a bunch of preclinical studies with pharmaceutical companies, and making sense out of this data is not trivial. So pretty much pharmaceutical companies will tell you that with every new technology that comes on the market that generates a lot of data, they need about 5 to 10 years to understand what that data really means to them. To give you an example on what it means in terms of throughput. So what you do today, if you do traditional H&E imaging and basically semi-quantitative automation for tissue microarray, which is like a little tissue stamp, you probably have up to 10 measurements. So if you have multiple fields of view, you get 100 measurements, and then you scale up until you have all sort of a whole multiplex. So the whole multiplex TMA already has 180,000 measurements. So a multiplex slide, which basically corresponds to this, has 18 million measurements. And now we should say you have a small study, 300 images per study, which is really not a lot. That doesn't even mean 300 patients. You go from 3,000 measurements to 4.5 billion measurements. Okay, and the question is now, in what information is in these 4.5 billion data points that was not contained in here? And we don't really have a definite answer to that today. Now, moving on to Oxford, there's now an institute, basically was just newly established, which is called the Target Discovery Institute. And what it basically has, it has 6 different groups. It's a relatively small institute. We have a high throughput screening facility. We have then paired with this quantitative imaging, epigenetics, which is like the assay development. We have a mass spec facility. And what the purpose of the institute is essentially bringing some of these high throughput screening technologies, like you saw on the previous slides, into the academic setting. Because there's really sort of a recognition that we want to scale up biological studies and also link these more towards drug development. And what you have, some of you might not be familiar with that setup. There's a group in Oxford which is called the Structured Genomics Consortium. These are essentially, it's quite amazing, these are sort of chemists that work in a fully open source way. So every time they have a new compound, they essentially give it to the world and the world tries it out. So the question is now, can we characterize these compounds in a sort of more systematic way and give other people that work and test these then in clinical trials, give them more guidance? So the idea here is, just to give you an example, is obviously we use imaging strategies to do phenotypic screening. We essentially want to make this process as easy as possible to sort of reduce the essentially all the sort of assay, all the sample preparation work. So there the technique of basically label-free microscopy plays a big role, and then we use fluorescent labels to then look at measurements like viability, differentiation, proliferation, and cell death. So why I'm showing you this is basically moving microscopy in general away from scientists that sit at eyepieces to essentially spreadsheets like you saw before. And the challenge is to basically develop image analysis or data analytics tools that are heavily automated. They are more automated than what you saw in medical imaging in the past, because in the medical image you could always say, if this is one exam, you can consult a clinician and get some input. Here you really want to avoid this because you want to generate a lot of data points automatically. And also the way the institute is now being set up, we now have Cancer Research UK Oxford Centre, which is sort of a comprehensive cancer centre, and you can view this as essentially people. It's fairly large. It involves clinical people, but it also provides sort of the experimental knowledge to sort of biological background to do something like targets discovery. I mean, we work in other disease contexts as well, but this is just an example. And then you sort of see how engineering feeds in by basically providing medical and computer vision, medical imaging knowledge. This is sort of my personal expertise. And the ultimate goal is, of course, to improve therapy, understand disease, and also discover new drugs. So now you basically sit at the other coin and say, look, if I want to understand a certain disease more carefully, then you have to develop very specific data sets. And now you go back. So for instance, we're looking at, I'm going to relate to this in a minute, but we're looking at, let me go look on this slide first. So this is sort of the question now. If you have a specific institute that sort of generates biological data and you have a big data theme, what are the current interactions, right? So I'm not going to go in detail through this slide, but what you want big data analysis and big data science for is also mine existing public repositories to inform what you do locally, right? Can you, for instance, generate really good hypotheses out of publicly available data? Can you then create models that are already guided by these hypotheses and then sort of feed back into the sort of big data analysis? And I think it will still take a while to sort of set up this sort of academic ecosystem so that it really works and is very productive. So finally, I want to go back to the theme of computational pathology for the following reason. Digital pathology is really something that's happening in the clinic now. You can sort of compare this to the advent of radiology, I don't know, maybe 20 years ago. And these images are also very large. They are probably 3 to 6 gigabyte per image. Obviously, an obvious example is already that pathologists can work remotely, but we also want to support the work of pathologists with sort of quantitative measurements. Some of you might be familiar with the fact if I grade certain cancer types, this is a largely qualitative assessment. This is really categorical. So there's a great opportunity of basically assisting that analysis with quantitative measurements. And then, of course, we want to use these kinds of data sources also in clinical trials and get more information out of those. So that brings me into the context of cancer immunotherapy, which is probably, well, one of the newer, very, very promising approaches to sort of use it as an additional tool in combination therapy. You know, it's been hailed as a science breakthrough in 2013. Patients that do respond really have substantial prolonged survival times, but there's still the question, what patient group does respond? So that's an open question. So we are currently designing a clinical trial that's specific to esophageal cancer. And now you're basically in the situation that you have to generate a data set that is really specific to the question you're asking. And we want to make use of tools like single-cell sequencing. And then all of a sudden, just gathering data from 60 patients costs unbelievable amounts of money. So you see the flip side of the coin. I wouldn't say it's easy to produce big data, but it's possible. But on the other hand, if you really are in a specific context and you look at what data do I really need, then it is very, very difficult to basically generate it and also to curate it. Imagine like for the level of really annotation and curation you need to make these data sets usable is quite large. Just a couple of slides on quantitative imaging. We're sort of working towards almost like developing a visual vocabulary to analyze, especially now this is in the focus of esophageal cancer. You basically have these different glandular types, can be extracted and can be categorized them. Pathologists have also done this. They sort of know what kind of types exist. We want to detect specific cell types and structures. And sort of in summary, really develop a visual vocabulary for tissue analysis. These are some of the early examples. It does involve a lot of machine learning. We essentially generate a map of the tissue and then index information into that map. So you saw parallels to the sort of tissue map I showed you before. And we basically want to assist pathologists with detecting specialized cell types and stuff like that. So that's already available even in some commercial products, but basically tying it all together. And then you also see that I spoke about single-cell sequencing information. You can basically index this into the tissue using these kinds of techniques. So looking ahead, robust algorithms are important, but really they're sort of pieces of a puzzle. And what we are moving to, in especially I think in biomedical applications, I call them sort of enterprise-level applications. So why are these enterprise levels? We basically we collect a lot of data in a sort of defined way. And we're really not analyzing 5 or 10 images, but we basically have a whole workflow where we apply these tools over and over again. Sort of an example here is at Genelia Farms that's near Washington. They analyze fly brains, and they can now analyze up to about 400 fly brains a day in a fairly robotic way. These are all things we haven't dreamt of maybe 10 years ago. We are starting to really link patterns across anatomical scales. We can mine this information, and then not many people have taught about this today, but really interacting and visualizing these data sets is incredibly important, because we can't just have black boxes where certain answers come out. That concludes my talk.